This experiment was performed to determine the concentration of an unknown acid solution through a process called acid-base titration. An acid-base titration is a procedure which is used to determine the concentration of an acid or base. An unknown acidic solution was used to neutralize a specific volume of the unknown concentration. It was neutralized by adding the base, sodium hydroxide, to the acetic acid, vinegar, then adding 2 drops of an indicator in order to balance the solution. We could determine when it was done neutralizing by the color change that resulted in a permanent pale pink. However, if too much base was added, the solution resulted in various darker shades of pink than expected. The average concentration that was promised was 12% of acetic acid, and our lab resulted in 11.7%. This means the acetic acid will result in a different taste. These findings suggest that the concentration was not the average 12% that was promised.
The titration of acetic acid lab was performed to discover the endpoint of titration. When performing the lab, we used the following equation to calculate the unknown concentration: volume of acid and molarity of acid, volume of base and molarity of base. The endpoint is indicated by the sudden and permanent change in color of the indicator used. In this case, the color that indicates the endpoint was a pale pink. The following materials were used to perform this lab: a burette, flask, distilled water, phenolphthalein, stirring rod, and a measuring tube. The burette was used to hold 0.1 moles of NaOH. The flask contained 5 milliliters of acetic acid, and 100 milliliters of non-distilled water was added to it from the measuring tube. 2 drops of phenolphthalein was then put into the flask. After stirring the solution, we added as much base required to turn the color a pale pink. When performing the lab, we found the initial and final volume of the base NaOH. In trial 1, the final volume we used of NaOH was 33.5 milliliters. By getting these two values, we were able to determine the volume of the base that was used. In the second trial, the final volume we used of NaOH was 32.9 milliliters. By using the final volumes, we were able to determine the molarity of acetic acid. In trial 1, the molarity was 1.90, and in trial 2, it was 2.01. Neutralizing was the main reaction in the acid-base titration. During neutralization, the acid and base reacts to form a salt and water. All the reactants appeared to be clear and would remain that way if an appropriate indicator was not added. Adding an appropriate indicator would result in the color change at the endpoint. The indicator phenolphthalein was used because it was a weak acid-strong base titration. The article acid-base indicator states, adding hydroxide ions removes the hydrogen ions from the equilibrium, which tips to the right to replace them, turning the indicator pink.